This is Microbiology Lecture 20. What we're going to look at now are microbial infections or diseases of the cardiovascular and lymphatic systems. Just to remind you, the cardiovascular system is a method for distributing nutrients, gases, metabolic waste throughout the body, and a few other things like hormones, and electrolytes, etc. There's a arterial side and a venous side to the circulation. There are two major circulations, the systemic and the pulmonary, and the heart is acting as a pump in between. Let's move on. The lymphatic system includes the lymphatic vessels that drain any excess interstitial fluid which originates actually from capillaries. It's almost acting like, in a sense, like a flush through your cell, through the interstitium, through the space between cells, flushing anything that's present in that space through these vessels that all lead back to the blood vasculature. The lymphatic vessels drain back into the left subclavian, They drain back into the left subclavian vein at the thoracic duct. That's what the thoracic duct leads into the left subclavian vein on the left side, and on the right side, the right lymphatic duct drains into the right subclavian vein. So all that lymphatic fluid transporting lymph, which was originally interstitial fluid, and anything that is dissolved in it, all leads back to the blood and enters into the venous system at the left and right subclavian veins. The lymphatic system is populated by white blood cells which originate in the bone marrow. The bone marrow and the thymus are both primary lymphoid organs. That means that they produce white blood cells of the white blood cells of the immune system. The bone marrow, of course, produces and is site of maturation of B cells or B lymphocytes, and the thymus is the site of maturation of T cells, T lymphocytes. All along the lymphatic vessels, you have lymph nodes that contain tissue macrophages, T and B lymphocytes, and these act as a filter and a site of where all the components that are required for a proper immune response to a foreign molecule and foreign target antigen are present, so that if there's anything foreign that drained into the lymphatic vessels, into the lymph from the tissues, that's where you often would get a response at that site in the lymph node. The spleen, on the other hand, also part of the lymphatic system. Lymph nodes, by the way, and spleen are secondary lymphoid organs, meaning that that's where the mature cells can carry out their function. So the spleen contains macrophages and T and B lymphocytes, and it helps to filter the blood. So it's monitoring the blood for any foreign target antigens that might be present. As I said, when the blood enters into a capillary bed, there is flow of some of the fluid out into the tissues, and that flushes through the interstitial space between cells into the lymphatic capillaries that act as a drainage, and that all head back up towards the left and right subclavian veins. And along the way, as I told you before, the lymph nodes that act as filters. Okay, so that's a quick review of lymphatic and cardiovascular systems. Now we'll go through different types of infection that can occur in those systems. Okay, Well, we should define certain terms. Sepsis is a condition of having bacteria growing in the blood, and that's associated with fever, chills, and increased respiration rate and increased heart rate. During sepsis, you typically have a lot of cytokine production. I remind you that these are soluble proteins produced by cells, different types of white blood cells, particularly T cells and macrophages that produce the kind of protective responses that normally occur with infection. Severe sepsis, you can actually get a drop in blood pressure, severe drop in blood pressure, and organ failure. And then more severe would be a person going into septic shock, in which low blood pressure cannot be controlled, and you start getting multiple organ failure. And that's usually, quite often, leads to death of the person. In terms of sepsis, gram-negative organisms, when they cause sepsis, the early symptoms are not particularly severe and are fairly nonspecific. They are due to responses triggered by endotoxin, causing a drop in blood pressure and cytokine release. Sometimes the treatment with antibiotics can initially just temporarily worsen the condition, because as the bacteria are killed and are dying, they're releasing the endotoxin or lipopolysaccharide from the cell wall, and so things get a little bit can actually get worse before they get better. In gram-positive sepsis, actually is often associated with nosocomial infections picked up in the hospital. Things like Staphylococcus aureus or Streptococcus pyogenes, also known as gas organisms. Sometimes group B streptococcal infections in newborns or in wounds. Enterococcal infections that are vancomycin resistant. All of these can lead to gram-positive sepsis. Okay, there's also, in terms of gram-positive organisms, puerperal sepsis is a problem, which used to be a huge problem before the advent of aseptic technique, understanding the fact that there are bacteria around us, and the development of the idea of using aseptic technique. Puerperal sepsis resulted often in very high mortality rate in obstetric cases, women who are, have just delivered a baby. Puerperal sepsis typically is caused by Streptococcus pyogenes gas organism, group A beta hemolytic Streptococcus. Usually, not often. Usually, not always, but usually. Often it's a nosocomial infection that the mother picks up in the hospital, and can be was in the past typically transmitted by attending physicians and midwives to the mother who was in labor. This has been tremendously reduced since that time. In terms of bacterial infections of the heart, they can trigger endocarditis and inflammation of the endocardium, the inner lining of the heart. Subacute bacterial endocarditis is often caused by streptococci infections from the mouth. Acute bacterial endococcus, also from Staphylococcus aureus, and pericarditis, which is inflammation of the pericardium, the lining in the heart, are also I'm sorry, that was Streptococcus for subacute and Staphylococcus aureus for acute. Pericarditis often is a streptococcal infection associated with streptococcal infections. Now, bacterial infections of the heart, the problem often is that you get clot formation, fibrin, platelet deposition on the valves, and that's what you see here in this diagram or in this photograph. You see the here a papillary muscle. These are chordae tendini leading up to a valve, which is completely encrusted with fibrin strands and platelets. Now, one of the problems of having streptococcal pyogenes or gas infection is that it can develop into rheumatic fever. Rheumatic fever is a disease process that involves cross-reaction between the immune response against Streptococcus pyogenes and the heart valves. Now you might be wondering why there's a photograph of an elbow here, but often one of the signs of that are these nodules that can form on the elbows. This is an autoimmune complication of Streptococcus pyogenes infection, and quite a serious disease process. Okay, we're just going to switch topics now for a bit to bacterial biological weapons. These are there's a long history among humans in attacking or planning on attacking, preparing, or doing even research into attacking others by developing biological weapons, which, you know, if the person developing them or using them wasn't also susceptible to it, I suppose it would make sense if you were, you know, trying to attack or defend against some aliens, but when you are also human and are also susceptible to the exact same bacteria, then it is just incomprehensible. Nevertheless, it has been something that has been used for centuries. Even in the 1300s, there are documented cases of there was a siege of a city, and the army that was surrounding the city had plague caused by Yersinia pestis. The bacterium Yersinia pestis causes the plague. And they would take the bodies and throw them over the city walls into the city to send, at least they understood enough about infectious disease that they could understand that if they tossed the bodies using catapults into the city, then the people inside the city under siege would start getting plague. And this did happen, in fact, during the siege of Kaffa, which is a town in the Crimea in what is now disputed territory between the Ukraine and Russia. During the Sino-Japanese War, there was a war between China and Japan. Fleas carrying plague were used. The U.S. Army, as other major powers, was doing research into using bacteria as biological warfare, and in the 1950s they even tested dispersal equipment, spraying equipment from planes by spraying this organism, which is mostly harmless really, over Los Angeles, and then testing to try and pick it up. They use this organism because when it grows on agar, it's red, and there's nothing else that looks like it, so you can easily detect it if you were sampling areas that had been sprayed. In the early 70s, an international agreement between a huge number of countries, there's I think over 180 countries now that have agreed to this, made possession of biological weapons illegal and research into them also illegal, although that did not completely stop, certainly. There was a Bacillus anthracis plant explosion in the Soviet Union in the late 1970s. There have been isolated cases of small groups or individuals using bacteria as weapons. There was an outbreak of Salmonella enterica in the northwestern United States in a small town that had been well, there was a group, a cult group near that small town, and the cult actually started becoming politically active. And the town was not responding well to that, and so they poisoned the local restaurant's salad bar with Salmonella enterica. That was in the 80s. In the early 2000s, Bacillus anthracis spores, which are extremely difficult to actually prepare in any large number. It takes a really highly sophisticated, skilled individual to prepare. They were distributed through the mail system in the United States and mailed to members of Congress and some newspaper organizations in the United States. Some people died. So, biological weapons have been around for a while. When the international agreement making biological weapon possession illegal was signed in the early 70s, the United States turned those laboratories into research labs. They handed them over to the National Institutes of Health, NIH, and they were used for the National Cancer Institute labs. And those National Cancer Institute labs are in that place. I'll talk about that a little later. The Weapons Convention in the agreement in the early 70s made possession illegal, and there was a multilateral disarmament treaty banning production at the same time. And there are over 183 countries now in the world that have ratified the agreement. No countries have been proven to possess offensive biological warfare programs, although 9 are still suspected of working on them. Some with good evidence, others with suggestion, more accusation than evidence, but still 9 countries are still suspected of actually carrying out biological weapons programs. As I said, when these are some organisms that have been used or studied as potential biological weapons. As I said, when the international agreement was signed in the early 70s, the biological warfare labs, and this is the back side of the building where they were carried out, these labs were given to the National Cancer Institute. This sphere is called the 8-ball, and it's a sphere that was used to suspend, it's huge, it's about 3 stories high, was used to suspend test animals in and then deliver, spraying in biological weapons. So they put in all different kinds of animals to see if they, would pick up the infection, because they were trying to develop delivery systems also. These labs were at a military facility called Fort Detrick in the United States near Washington. What you should understand is that these, the larger countries, even if they have stopped doing biological warfare research, they are nevertheless researching highly infectious microorganisms and are able to grow them and keep them. They have them in culture and are growing and keeping them. in order to try and develop vaccines against them, protect against having them, and because there's no guarantee that someone else wouldn't try and develop them as a weapon. And that kind of work is done at Fort Detrick in a large institute called the United States Armed Medical Research Institute into Infectious Disease, or USAMRIID. And here's an individual working in an area with some highly deadly. They have at USAMRIID, they have every possible deadly thing you can imagine. Every kind of bacterial or viruses that can even potentially act as weapons. Well, which brings us to highly infectious organisms like Bacillus anthracis, which are endospore-forming rods. They're found in the soil. All cattle are routinely vaccinated because they're particularly susceptible to anthrax. It can reasonably easily be treated, but it is a serious disease, particularly depending upon the portal of entry. There's cutaneous, gastrointestinal, or inhalational. You might be wondering why I'm talking about this. It's in here because it has been used as a biological weapon, and this is the organism that endospores were prepared from and mailed to the United States Senate offices or Congress, United States Congress offices of elected officials back in 2001. Inhalation of the endospores results in, and doesn't take that many, results in 100% mortality, whereas gastrointestinal, if you eat the spores with undercooked food or contaminated food, it's 50%. Cutaneous is only 20%. So this is actually quite deadly. Okay, let's talk about tissue injury and the kind of injury that you get where there's a problem with the vascular system. If tissue loses blood supply, it becomes ischemic. It has inadequate delivery of oxygen. And because of the structure, shape of the vascular system, where you have, artery leading to smaller arteries, and then arterioles leading to smaller arterioles, which then lead to a capillary bed. If you have an infarct, a block in somewhere along there, then you'll end up typically with a wedge-shaped region of dead tissue. So say there's an artery leading to some arterioles, and the arterioles get smaller and lead to different areas, and then eventually you get to a capillary bed, which is the place where the gas exchange occurs. And that capillary bed eventually, so you have gas exchange, and then these join together to form into a venule, and then you get let me just show you again, and then you get into a venule leading to larger veins, etc. So you get this, if there's a blockage, say here or here, then you would, if there's a blockage here, then what you would get is typically a wedge-shaped area. I'm going to outline it in black. A wedge-shaped area, because the circulatory system is, fans out, and a fan shape like that in 3 dimensions is the same shape as a wedge-shaped area. So typically, if you have inadequate blood supply into an area, there's a blockage or some problem, then you get a wedge-shaped region of dead tissue, and that is called a infarct. and the tissue dies due to the process of necrosis. If you have necrosis and death of tissue due to inadequate blood supply, I mean, there's many reasons why tissue might die, all of them leading to necrosis, but when it's inadequate due to inadequate blood supply, it's the result is gangrene, death of tissue due to inadequate blood supply. So ischemia is a problem with vascular infections or blockages, and it's worthwhile knowing these terms: ischemia, infarct, necrosis, etc. Now, there is a organism which causes gangrene, and it's called gas gangrene because there is production of gas associated with the infection in the tissues. This organism is an endospore-forming anaerobe, which we've talked about many times. Is Clostridium perfringens. Typically grows in necrotic tissue because necrotic tissue does not get adequate oxygen supply. The vasculature is damaged and the tissues aren't receiving proper oxygen. Treatment is by removal of all necrotic tissue and sometimes put in a hyperbaric chamber even. That's a high pressure chamber with an oxygen enriched atmosphere because the organism is actually, oxygen is toxic to it because it's a obligate anaerobe. This is the kind of organism that causes infections and problems typically in wounds that are contaminated with dirt and feces that you might find on a farm, deep penetrating wounds. Okay, those were organisms, bacterial organisms. Now let's talk about viral infections, the lymphatic and cardiovascular systems. The two of the viruses that can infect these systems, which of course are connected together, lymphatics drain into the cardiovascular system. Two organisms that can infect them are human herpes virus 4, HHV4, more commonly called Epstein-Barr virus or EBV, and HHV5, more commonly called CMV or cytomegalovirus. I remind you that HHV1 is herpes simplex virus 1. HHV2 is the same as herpes simplex virus 2, which cause cold sores. HHV3 is the same as varicella zoster, which causes chickenpox and shingles. And here we have HHV4, that was 3, and HHV4 is Epstein-Barr virus, and HHV5 is cytomegalovirus. These are found infections in the cardiovascular system. HHV4 or Epstein-Barr EBV, Epstein-Barr virus, is transmitted via saliva. Sometimes called it causes a disease called kissing disease for obvious reasons. The severity of the disease increases with the age of the individual. In childhood, the infections are asymptomatic. This is something that people should be more aware of, that children can get infectious diseases and be completely asymptomatic and yet spread the disease, but they have it and they can spread the disease. Young adults, teens, get, if they're infected for the first time with EBV, they'll get a disease called infectious mononucleosis. As I said, also caused kissing disease, which is characterized by a proliferation of monocytes in the peripheral blood. Older adults, the infection has been clearly associated with a type of cancer, nasopharyngeal carcinoma, more common in Asia than in the rest of the world, and also Burkitt's lymphoma, more common in Middle eastern side area of the African continent, but can occur in other places. This virus can cause cancer in immunosuppressed individuals and is a problem in AIDS patients who are immunosuppressed. I don't know why it says and malaria. It doesn't cause cancer and malaria and AIDS patients. Oh, malaria patients, and in malaria patients and AIDS patients it can cause a type of cancer, the EBV. In fact, Burkitt's lymphoma is associated with co-infection with the malarial organism and EBV at the same time. Here you see some graphs. This is from the United States in terms of ages of individuals and how many are positive, how many have antibodies showing that they were infected at some point with these different viruses, EBV or CMV. I should point out to you that by the time a person reaches 21, they have a high incidence of a high probability of having been infected. In Quebec, more than 80% of people by the time they reach their early 20s or mid-20s, more than 80% have had CMV exposure and EBV, both EBV and CMV. They're positive, they show antibodies. Okay, let's go to human herpes virus 5, cytomegalovirus, a little bit more detail. It causes a swelling in cells. It can remain latent in white blood cells. The person can be asymptomatic or have a very mild response. Transmitted across the placenta into a fetus can cause neonatal damage. It can cause fetal damage, resulting in neonatal neural damage. I think that's about it for that. There are a large number of viral hemorrhagic fevers, and some are transmitted by insect vectors between animal reservoirs and humans. They can infect animals, but they also sometimes get carried into humans. And the one that and others don't have an insect vector; they directly will infect humans. The one that stands out, I suppose, is Ebola virus. The host could be monkeys or bats. A much feared, terrible, rapidly progressing viral hemorrhagic fever, causing bleeding from many sites. There you see some Ebola virus particles. These long strands are Ebola virus. Well, one of the most well-known vector-transmitted diseases is among the most well-known is the plague, also called the bubonic plague, also called the Black Death, and also called La Peste. It's a infection caused by a bacterium which used to be called Pasteurella pestis, but the name was changed, I don't know, maybe 10 or 15, maybe even 20 years ago to Yersinia pestis, based on DNA sequencing. It's a bacteria that is normally carried quite often in prairie dogs and ground squirrels, believed to be endemic in Middle Asia, in the Mongolia area, but spread towards the west and spread throughout the world. Also carried by rats. The organism is moved from between animals, between host animals, by a flea. The growth of the organism in the blood and lymph results in bubonic plague. If it gets into the bloodstream, it can induce septicemic shock or septic shock. If it's in the lungs, it causes pneumonic type of infection. But the one that's epidemic and is most was a huge problem, and at times killed, I think, something like a quarter or a third of the population in Europe, was the bubonic plague. It's found actually among I think it's ground squirrels or some kind of rodent in the southwest United States, and there's always a few cases that occur in humans. The name bubonic plague refers to these swellings that you find on the people who have it. They get this kind of swelling of the, shown here, swelling in the lymph nodes so bad that it seen at the surface and they turn necrotic and black. Quite terrible. Okay, another vector-borne disease that is actually still present and even growing in incidence is Lyme disease caused by Borrelia burgdorferi. There's a mouthful for you. It's a bacteria which in which the normal host, a normal animal that carries it, is deer. It is transmitted between animals and to humans by ticks that take a blood meal off the deer, and then when you're walking in the forest, particularly in the fall, you can pick up a tick and get a tick bite. For those of you who, well, here's the life cycle. This is actually, we're not going to get into the details of the life cycle, other than deer normally have the bacteria, and ticks pick it up, and then can, it can also involve rodents. I'm going to go on. For those of you who aren't familiar with ticks often will take a blood meal. This is a pin to give you an idea of the size of the insect. It's quite tiny, but when it takes a blood meal, then it swells up quite huge, and you're not always aware that it's on you. You'd think you would be, but you're not aware it's on you, and it's taking a blood meal. It can swell up to the size of a small grape with blood. The organism, the bacteria that actually causes the disease, is Borrelia burgdorferi. One of the first symptoms is it goes through different phases, and an early symptom at the site of the tick bite is what's called a bullseye rash. And the idea here is bullseye refers to a sort of a circular target kind of a appearance, right? You get a darker red area around a lighter area in the center. After that, the rash can disappear, but in the second phase, you can have effects on the heart and inflammation of the brain. And the third phase is arthritis. It's a difficult disease actually to get rid of, to treat. And anyone who goes hiking, particularly in the fall where there's deer populations, and that certainly is here, should be careful that they don't pick up any ticks on them, on their body anywhere. The tick actually, when it takes a blood meal, defecates, and that gets into the wound, and that's what spreads the disease. Another vector-borne disease is epidemic typhus. It's a disease caused by members of the genus Rickettsia. The animal reservoir are rodents, and the insect vector is lice, body lice. And transmitted with lice feces that get into the bite wound. Epidemic murine typhus. Let's go on. There's several different types of epidemic typhus diseases. Spotty fever. I'm not going to get into this details of this. This is a protozoa or protist disease. Carried it's Chagas disease. Again, I'm not going to get into the details, but it is insect vector borne. Toxoplasma, again, insect vector. This is an important one. Again, a protozoa. Now we're on talking about protozoan diseases that can affect the blood or the cardiovascular system, blood or lymph. This is malaria caused by the protozoa Plasmodium. That's the genus name. The two most commonly associated with the disease in humans species are Vivax and Plasmodium falciparum. The vector in this case is a particular mosquito. And we've discussed it before. A diagnostic feature of it is that when you look at a blood smear, you see these ring forms inside the red cells that almost look like a nucleus, although red cells don't have one, so it stands out. And these are immature forms of the protozoa. Malaria is a huge problem in many parts of the world. It used to be much more widespread, but is still present in many areas endemically. You can see in Latin America, South America, Africa, through Asia, all these areas in purple still have it. It's although it used to be much wider, amazingly even into Canada, southern Canada. We associated mostly with the tropics, but it used to be much further north even. Really a huge problem. People don't realize just how many cases there are. We're not going to get into the life cycle, other than that you should know that the disease is spread because of the mosquito carrying it in the salivary glands, and when they bite human, they will always inject a little bit of saliva, and that's how the organism gets into people. In 2016, there were approximately 216 million cases worldwide. Giant disease. Still endemic in these areas that you can see colored in here. Estimated somewhere between 400 and 700,000 deaths per year. Just a gigantic problem. Another protozoa that infects humans, which is a problem, is and it causes the disease leishmaniasis. This is again a protozoa or protist. There are 20 different species worldwide that can infect humans. Vector borne by sandflies, 4 to 12 million cases per year, up to 50,000 deaths annually. So here are the different leishmaniasis types. Quite a few different species of the organism. I'm not going to get into all of it at all this detail. Another protozoal disease. These are all, malaria, schistosomiasis, leishmania, all serious diseases. This is a parasitic disease, schistosomiasis. It's a flatworm or platyhelminthes. Snail is an intermediate host. Disease is also called in some parts of the world bilharzia. The snail carries it. People walk in the ditch in the water, irrigation ditches in fields, farming, and pick it up. It burrows into the skin. Approximately huge, 250 million cases worldwide, up to 200 deaths estimated per year. It's debilitating and affects a huge number of people. What this organism does is it gets in and then it burrows into the muscle tissue and insists in there in the tissue. It has an unusual association. The male and the female flatworm live together essentially. Always good if you're a parasite to have your sexual partner right there right away. I mean, as you know, parasitic worms are completely evolved to reproduce and shed eggs. Schistosomiasis is also associated and present in Canada. As you can see, the snail is carrying it, and these immature forms called cercaria get released, and they will burrow, as I told you, into the skin. In Canada, what they do is they burrow into the skin, but they don't actually permanently infect people, but you do get an allergic response in your skin, and that allergic response is called swimmer's itch. And you can see this is a table showing different types of schistosomiasis worldwide, but these actually do infect and get into, here the urinary bladder, intestinal wall. Here it's just in the skin, swimmer's itch, and this is a map, fairly up-to-date map, where cases occur in Canada. People reporting cases of swimmer's itch in Canada. In this case, the cercaria, immature form of the flatworm, tries to burrow through the skin, and you get a cutaneous allergic reaction. It's carried in North America in waterfowl, ducks, geese, wild ducks and geese, and gets into the water system, into the snails, and then the cercaria come out of the snails, and if you're in the water, and you can get the immature forms trying to burrow into your skin. Although human is not a normal host, they do try to get in through the skin, and you get this allergic reaction and resulting in swimmer's itch. Food for thought, huh? Okay, so that's the end of lecture 20, microbiology.